Okay, great. Looks like I'm on the screen. Okay. Can people at home hear and everything? Is that working? Hopefully. Oh yeah, I see some thumbs up. Okay, cool, great. All right. Let me get my pointer out here. Well, thank you all for the invitation to speak with you today. This is my first trip out to Silver City, New Mexico. I actually hadn't even been through Hatch before today, so I'm on an adventure. I'll introduce myself a little bit at the beginning of this talk. How many of you were at the annual Native Plant Society meeting last summer in Albuquerque? Just a couple, okay. So it might be a little bit redundant for you, my little introduction, but I thought since I'm relatively new to this state and to the community that it would be helpful, to kind of show a little bit about where I came from to get here. But otherwise, what I want to do today is to kind of introduce to myself, but also a little bit of my research interests and what I'm doing here in New Mexico with my lab group, as well as the Herbarium group, to try to get a better understanding of the biodiversity of alpine ecosystems in particular. I'll be talking about alpine plant communities kind of broadly across North and South America at first, but then get into some more of the details about really how we're trying to understand specifically the Southern Rocky Mountain alpine flora. This flora is one of the least studied floras in North America for probably reasons that you can understand, right? It's a pretty remote area to get to, a lot of challenges collecting plants and getting once you get out to these areas. So, things like the monsoon storms that are already picking up this year, right? So it can be a sort of a dangerous place to collect plants as well. But once you get out there, there's these really spectacular plant communities, which I really love. So I'm going to jump right in and start with a brief introduction of myself. Let me see if I can, I don't know if I can show you this thing at the top. It's not that big of a deal, but
I've had problems doing it in the past.
yeah, I'll just leave it. But basically, I arrived, my journey to my interest in trying to understand alpine plants. Let's see here, there we go. Started at the University of Washington. So I grew up in Washington State, Southwestern Washington, in a suburb essentially of Portland, Vancouver, Washington. And then I went to school at the University of Washington. But when I started school, as I was sharing with some of you at dinner, I was actually a dance major. I knew nothing about plants. I loved biology, so I really liked science. I knew that. And I was thinking, maybe after my career in dance, I would go into like some type of health science career, right? Become a doctor or a nurse or something like that. But as an undergrad, I spent the summers working in central Idaho at a mountain lodge, trying to pay my way through school. And in my free time, so I was basically a housekeeper there. And in my free time, I was doing a lot of hiking around outside, and I had a lot of free time. And I got really interested in all of these amazing alpine plants that I was seeing, but I didn't know anything about them. So I went back to school in the fall and registered for a plant identification course. And I had this really spectacular teacher, Richard Olmsted, who's an expert in the Lamiales group. And he just totally changed my career path. So I got really interested in botany and trying to understand specifically how plants are related to each other. So I got into his molecular systematics lab as an undergraduate and started working with him. And it just kind of was amazing to me that there's still so many questions that we have just about basic biodiversity of plants, plant life on this planet, So I took a few years off before I decided I wanted to go back to graduate school, and was doing some vegetation surveys in Central Oregon and Three Sisters Wilderness. And there I started getting even more interested in understanding ecological diversity. So as an undergrad, I was kind of looking at evolutionary relationships among plant species, but then I started thinking, well, I was really fascinated about how, the plants that we're seeing out in ecosystems change, and why do they change, even within like a few hundred meters of each other and over, elevation gradients and these types of things. So I decided I wanted to go to graduate school and pursue some of these questions. So I ended up at the University of Idaho. Partly because that's kind of where my whole interests began, right? So I started a PhD dissertation trying to understand how ecological plant communities change and why. So I spent some time actually looking at species invasions, particularly on the San Juan Islands, which are this amazing archipelago off the coast of Washington State. Many of you guys might be familiar with it. What you might not know is there's about almost 500 islands in the archipelago. Some of them are very small. Some of them are only a few meters in diameter. But despite this, about 30% of that flora is now invasive. And so we were looking at patterns across the islands, but I also started looking at alpine plant community diversity and comparing the diversity of alpine plants on summits in central Idaho. One part of my graduate education actually brought me to the French Alps. So I also was able to have this really cool opportunity to compare patterns of alpine biodiversity between the French alpine summits and the Idaho alpine summits. So I started getting really interested in this idea of trying to link together all of these different patterns of biodiversity across larger spatial scales. So I graduated, I received my PhD, and then I went on to pursue some additional training as a postdoctoral fellow. And so I spent a couple of years at the University of Arizona, and there I was actually learning some special genomic skill sets that I was trying to apply into my research program. But I also started to just really fall in love with the Southwest there. So I hadn't really spent much time outside of kind of like rainy, gloomy climates. And then all of a sudden I was living in Tucson and I was like, oh my gosh, the sun is amazing. I feel way more energized and I love the culture and stuff. So I had in the back of my head that I was like, man, it'd be really cool to end up back here someday. But before, I had another postdoctoral fellowship for 2 years in Michigan, at the University of Michigan. And the goal of that project was to work on actually exploring alpine biodiversity along the Andes. However, halfway through, COVID hit, and so that project kind of changed directions. And also about the same time, I was offered this position at University of New Mexico. So I was offered the position in 2019, deferred for a year, and then COVID happened. And so I started actually in January of 2021. So I've been at University of New Mexico for just over 2 years now. So I still feel really new to the area, especially since for about the first year I was here, everything was still shut down at Albuquerque. I don't know how it was down here, but it was very, very insular. I was teaching remotely for the first, I didn't meet most of my colleagues until the fall after I arrived. It was crazy. But a lot has changed. And now I have this wonderful group of people working with me to try to understand patterns of plant diversity. So this is a picture of my current lab group, as well as people who work in the herbarium. So I'm an assistant professor in the biology department, but also curator of the herbarium at UNM, and we're part of the Museum of Southwestern Biology. So we have this really great group of like museum-oriented folks out there. A lot of you may have interacted or somehow gotten an email from Harpo Faust. Harpo is our amazing collection manager. Phil Tawney, who some of you may have known or met before, retired a few months after I arrived. And so I was able to hire Harpo, and Harpo has really breathed like a lot of energy into our space, and it has really helped me to recruit a lot of these other students who are now working with me. So I have a graduate student, my first PhD student, Joseph Kleinkampf, whose work you'll hear about a little bit later in this talk. I have a few different undergraduates working especially on this alpine biodiversity project. I have a postdoc, Lizzie Lombardi, who I'll also be sharing some of the project she's working on, and two master's students who joined last year, who are doing really great things. Brianna is actually working on a floristics inventory of the Brokaw Mountains, and they're actually in the field right now. So that's pretty cool, another very underrepresented area of the state. And Erin is looking at, is comparing historical herbarium collections that have been collected in alpine areas with collections that she's making now to see if we can detect shifts in the floristic composition over time. So I'll kind of come back to that at the end as well. But yeah, I wanted to make sure to spend time introducing them because a lot of the work I'm going to be talking about today has really been led by a lot of them. And so it's just been a pleasure to be able to have this group already formed in a couple of years. Okay, so just kind of general big picture, what I'm interested in my research is trying to understand, why we see this incredible diversity of species on our planet, and how this diversity and why this diversity changes, again, like over space, so between ecosystems or elevation gradients, but also over time. And so, using the collections that we have available to us has really been invaluable for addressing a lot of these basic questions I'm interested in. So I hope that that's like the big takeaway that you get from my talk today. Of course, I'm most interested in the plants on this slide. I just somehow got really obsessed with botany. And I think, again, it goes back to having this really great teacher as an undergrad. But what I'm trying to answer is what types of big picture processes, both ecological, so species interactions type of processes, but also evolutionary processes. So where these plants may have evolved from back in time and how they've migrated into different areas, and how these interacting forces are driving the diversity and shaping the biodiversity that we are observing today. And as I've mentioned, I've become particularly fascinated in alpine ecosystems. So this map here is highlighting in black areas on our planet where we have what we consider to be alpine ecosystems. And you may not know, but this type of ecosystem is really found on every single continent on our planet. And you can see surrounding this map, just sort of a snapshot of the plants that you might encounter in these areas. The definition of what is alpine is actually something we could maybe debate a little bit. It's kind of a fun thing to think about because it's often defined by the vegetation itself, right? So this is a quote that I actually really like that defines one definition or describes one definition of the alpine that's taken from Edward Cassiter's Vegetation in Mexico. And he describes it as a treeless zone at and above the level of the last dwarf Engelmann spruces, subalpine firs and bristlecone pines, which is characterized by low, often matted vegetation of hardy alpine plants, some of which occur on the Arctic tundra of the far north and reach their southernmost limits on the New Mexican peaks, right? So I like this quote because, again, it kind of defines this ecosystem by the plants that live here. And he also is pointing out some of the kind of characteristics that we observe across many diverse species in the alpine. So we often see these kind of low cushion type of life forms, like these matted life forms. And he's also kind of describing a little bit about the extent of the alpine, right? It's very broad, as you could have, as you noticed on that previous map. But we do have alpine peaks down here in New Mexico, right? And I like to think of the New Mexican alpine summits as being sort of the forefront of the alpine, the beginning of the North American. So, very broadly speaking, again, kind of referencing back to that Katstetter definition, in my research program, we specifically define the alpine as anything above tree line. Okay, so you can kind of see anything above this this pink line here. This is a photo that was actually taken by my graduate student Joseph this last summer on one of the summits that he was sampling. And I think it was one of his. I can't remember. I should probably not even guess because I know I'm probably gonna be wrong. But this is what we're talking about today. So plants that live on the tops of mountains. So these plant communities are at high elevations. And so they're really defined by environmental extremes, right? They're at the highest elevations. Plants in these ecosystems experience long periods at freezing temperatures, extreme ultraviolet radiation, right? Because the sun's constantly beating down on them, and also long periods of drought. So for most of the year, when snow's on the ground, water is trapped in that snow and not easily accessible for most plants. Growing season is short, right? It can sometimes be just, a month and a half, 2 months. So a really short period of time for these plant species to complete their entire life cycle, right? So that's pretty incredible. They're often considered to be sort of island-like. So here's a photo I actually took from a plane window flying over Canada somewhere. And I like it because I think it kind of really illustrates this concept of alpine summits looking sort of like sky islands, right? So there are these kind of discrete areas, which are nice for kind of evolutionary and ecological questions because you can define them, but they're also separated from one another. So there's different challenges for, dispersal and migration and these types of things. Of course, they are also oftentimes very challenging to access, right? Challenging to get to study because they're remote and also environmentally extreme areas. But despite, all of these sort of physiological challenges that plants have to face, we still do know, especially from areas that have been better studied, that mountain summits actually promote or host a pretty high diversity of species. So a pretty high number of species richness. So here's sort of a snapshot of what you actually might encounter on only one summit. So this is actually a poster that I designed during my PhD research in central Idaho, showing a snapshot of alpine flora on a summit in that mountain range, right? And so you can see there's just a really incredible diversity of plants, not just, in terms of color, which I think maybe is one of the more striking things here, but also in the, the life forms, the type of root systems that these plants have, these types of things. So the big question is, why do we see so many different species of plants, such diversity of plants living on these mountain summits if they're trying to, withstand and survive in such an extreme environment? And of course, the reason that I think most of us are interested in understanding biodiversity in the first place is trying to see if we can detect patterns in what's shaping it so we can somehow understand how they'll change in the future. So today I'm going to talk, says on the top, documenting biodiversity. I'm going to talk about a couple of different approaches that my lab and I are using to try to better understand the diversity of alpine summits generally, and really, specifically moving forward in the Southern Rocky Mountains. So there's a couple of ways, broadly speaking, that we're trying to describe or document this biodiversity in the first place. And the first is by looking at collections that we already have. So looking back at historical collections, these collections are often, for the most part, housed in herbarium cabinets, right? And our herbaria hold these really incredible repositories of biodiversity. We can look to these past collections to understand, how biodiversity has changed over past spatial scales. But we can also actually take samples of these old collections and get genetic information out of them to try to understand from more of an evolutionary perspective how these communities are changing. So looking at where we have collections and where we don't can also try to inform these dark spots in our understanding of biodiversity across space and point to places that we might need to visit to try to fill in those gaps. Another approach that we're taking is adding new collections. Right. So, going out to places maybe informed by our historical collections, we can try to fill in some of these collection gaps. So we can, for instance, in the alpine, target certain summits that have never been collected before, right? Or, from more of a taxonomic component, we can try to focus on specific groups of plants that are understudied. We can also resample areas that have been very well studied. This is really important, especially for trying to see how communities change over time, right? So we can kind of, look back to historical collections to, again, inform new collection efforts. With new collections, we can also try to implement more sort of modern or updated collection practices. So a lot of this has to do with adding other information that we kind of wish we would have had, right, in our historical collections. So certain things like the specific location where we collected plants. A lot of times we try to go back to find a community that has been, documented before, but because the locality of where it was collected said, somewhere near Santa Fe, we really don't know where that is, right? But now we have this amazing technology. We have satellites in the sky that we can put a real precise locality on these plant collections. We can also collect some tissue of the plant, specifically leaf tissue in silica. And silica is, silica powder is like this desiccant, right? You get the packet in your new tennis shoe boxes. It wicks the moisture out of the plant very, very quickly. So it dries it out very quickly. And this makes it much easier to extract genetic information from that plant tissue. So we're trying to kind of implement these new collecting techniques as we move forward and document biodiversity. And so this is sort of a big initiative that we're working towards at the UNM Herbarium. So both looking, in our cabinets to see what we have, using these historical collections to inform places we want to go, and at the same time trying to update our practices for the future. So I'm going to start today with kind of, walking you through some examples of how we're first using historical collections to understand biodiversity or the state of biodiversity in alpine communities. And then I'm going to end with some examples of how we're trying to either fill in these gaps or complement the knowledge we have from our historical collections with new collections to try to understand biodiversity from that perspective. So first, looking at the collections that we already have. So how many of you have been to an herbarium before? Have you all been to this herbarium maybe, hopefully as part of, part of this? Okay, great. So you're familiar a little bit with what an herbarium sheet looks like. That's good. I don't have to spend much time with that. But, and you also know there's a lot of information on this sheet, right? Besides just the physical specimen, the pressed plant, there's a lot of associated information about where the plant was collected, when it was collected, who was collected by, right? The accuracy of this is getting better and better over time, but it still is an amazing wealth of information. And most herbaria now have been working towards trying to make this information on the physical specimen more broadly accessible in a digital format, right? Have you all used SciNet before? Heard of SciNet? Okay, a little bit. So this is a screenshot of SciNet. And SciNet is this resource again, that most herbaria in the Southwest are using to try to move all this physical information into a searchable format. So at UNM, most of our collection has been digitized. So we have over 130,000 vascular plant specimens. Over 80% of these have now been digitized. So that means all of the collection information of that specimen, but also an image of that specimen in high definition is available. So you can go to sign it and search for a plant, and you can see the actual photo of that actual specimen. This is really an incredible thing if you think about it, because we are only one of almost 5,000 active herbaria around the world. For the past almost 2 decades, herbaria have been working towards this digitization of their collections. Some of them are further along than others. We were fortunate, we received funding from the National Science Foundation, from the government, to digitize our collections. That's why we're so far along. Other places like Kew Botanic Garden in England actually have just started their efforts. So, they have millions of plant specimens. So once that gets going, it's going to really change things a lot. But this map is just showing the distribution of where hotspots of digitized effort of collections have been. So you can see, in the US, we've been doing a lot of digitization, again, thanks to funding from NSF. It's been amazing. In other areas of the world, right, notably Africa, there hasn't been as much of a push, but things are moving forward. So this map is changing. So from these digitization efforts, we have, over 380 million specimens worldwide. Like, that's a ton of information if you think about it, right? So what this looks like is some of these local, so like UNM, Herbarium, we publish our digital information on sort of a local database called SciEt. And so you can Google that. It's an amazing resource if you haven't checked it out before. There's a lot of things you can do on there that I don't have time to get into today, but I'd be happy to talk about. Then all this information from these like kind of local digital resources gets added to increasingly more inclusive and larger repositories. So GBIF, you may have heard of, is a very large biodiversity database. And basically it just pulls in all of this information into like one platform that's easily accessible, and you can work with it more easily at larger scales. And this graph here is just showing the number of occurrences in millions that have been accumulating over time, right? So again, just highlighted, we have almost 400 million occurrence records now in GBIF. And this is representing like the known biodiversity of life on our planet, right? This is just for plants too. So, think about that. If you, consider arthropods and mammals and birds, we have so much information to work with. So this digitization of collections, not just plants, but other specimens, has really revolutionized science. It's made biodiversity information publicly accessible. So a lot of people like to think of it sort of democratizing science in a way. The uses of our collections now have really expanded beyond like taxonomy and understanding biogeography and species distributions to so many more things, right? So obviously lots of floristics, but also a lot of like ethnobotany and DNA analyses and things like this. So this whole field of kind of what I'm going to be talking about today, a lot of what we call biodiversity informatics, has just really been skyrocketing over time. So the first kind of example of how I'm using all of this biodiversity in my research to understand, again, these alpine plant communities is this project I worked on with a group of students, in particular, Hector, who was a graduate student at the time when I was at Michigan. So our field work in the Andes got canceled, sad, but instead we started working with these biodiversity data more. And so in this project, we looked at all of that information from these global databases, and we pulled out records of plants that had been collected only in mountain areas across North and South America. We used some modeling to try to get a better idea of how these species might be distributed across space based on where they were found. And we mapped this all out. So this map here is showing some of our results. So it's highlighting hotspots of species richness. So the number of species. So these warmer tones are indicating areas where we think we have more species, and bluer tones are indicating areas where we have less species. And so what it illustrated to us was, not surprisingly, this plant diversity is not evenly distributed across mountains. So despite the fact that, plants, kind of going back to the beginning of this presentation, have a lot of the same challenges, like physiological challenges living in these extreme environments, we have different species across space that we find. The central Andes have been known to be a hotspot of alpine plant diversity for a long time. There's been work on specific groups of alpine plants coming out of the central Andes, but we didn't really recognize the importance of North America, especially going up into Canada for alpine biodiversity. So that was something that really came out of this work and looking at these collections data. Looking at just sort of the number of collections that we have, so taking out the fancy modeling we did, but just looking at where we have numbers of these plants that have been collected, that's what this map is showing here. And in this case, still the warmer tones are indicating where we have more collections from. So what this is showing is that we have more collections from, Mexico and the northern Andes, as well as the West Coast in California. But there are areas specifically, in North America, right, that have very few collections. So these spots of red, I'm zooming in here to North America, are areas where we have no collections at all. So of course, a lot, in very remote parts of Canada, Alaska, but still we have some of these sort of dark zones of biodiversity in the Rocky Mountains as well. Again, not surprising, as we all know these areas are remote and challenging to get to, but this is just showing an example of how looking at spatially where these collections have been made can help to inform maybe where we should try to focus collecting efforts in the future. And so kind of building off of this, a project that my postdoc Lizzie is working on right now is trying to summarize the collections data that we have specifically for New Mexico to identify priorities for future collecting efforts and to identify in a way that's more informed than just, yeah, we have something here and not here. So we're trying to, first of all, see what biases exist in collections that have been made in New Mexico. So biases in space and time and taxonomic group. And also then given these biases, how can we look at them and think of ways to direct our future collecting work? So again, we're using collections data from these database efforts. There's a lot that we're doing here, so I'm gonna not go through the methods, but I just kind of wanted to give you a big picture overview. So using these diversity collections, we basically clean them up. There's a lot here. Taxonomy is a mess, if you can imagine. People typing in names incorrectly, lots of things that we have to do. But then we take all of the map data, the locality information, and map these collections in space. And we pull in additional information about different climatic zones, different ecoregions, and also socio-political areas. And so what I mean by this is we're comparing collections that have been made in public lands versus private lands. We're also pulling in maps from like Forest Service and BLM and comparing collections that have been made on BLM versus Forest Service. To try to quantify in different ways the diversity that we have across these different types of spatial areas, right? So we're hoping what this project can do in the end is then allow us to kind of compare across different interests, right? If we're interested in maybe looking at the alpine, for example, and understanding the number of collections that we have there, we'll have that quantification. And we can compare it in terms of like the spatial area of these different places as well. So this work is still in progress, but we have been mapping, first of all, the collections. So this is showing a map. The red dots are indicating collections that have been made in New Mexico. The color underneath them is just kind of summarizing the density of those collections in space. And so darker blue areas are showing hotspots of collecting efforts. Just generally, not surprising, concentrated around cities and roads, right? So that's what we've known that for a long time. We expected to see that, but this just visualizes it. We also, this is showing the number of different species that we have across our collections. So we have a lot of species with only one or a few collections that have been made, and very few species that have been collected multiple times across space, right? This is also a pattern that we see a lot, right? People tend to try to capture more the general biodiversity, and so you end up getting just a few of a species. But it's concerning if you're interested in understanding, how these species might be changing over time, like comparing traits that they have in the past, right, with the present day. If you don't have a lot of collections that you compare with, you can't really do those types of, answer those types of questions. We also see a lot of Asteraceae, right? I heard you had Rich Spellenberg here recently. There's a lot of asters in New Mexico, not surprising. And then, of course, grasslands, a lot of Poaceae and Brassicaceae, and then Cactaceae. So, just kind of illustrating, again, kind of what we know a little bit. So this is where I'm going to show you kind of an example of what we're trying to do with dissecting the different types of spatial data that we could look at. So looking at either socio-political or ecological in this case. So this is an example where we looked at the level 4 ecoregion definitions. So these describe different ecological areas in a certain way. So we can look at the number of collections within each of these ecoregions by the area of that ecoregion, right? So the map over here is just kind of highlighting how, some of these ecoregions are patchy, right? But we can still quantify that area. And so what we can see is that certain ecoregions have much more collections, especially, given their area. So these are mid-elevation forests. We have a lot more collections at mid-elevation forests that is proportional for their area. It's also highlighting places where we have, fewer than expected or, I guess, collections that we would like given the size of that ecoregion. So this is the Chihuahuan Desert ecoregion here that are highlighted. So makes up a big, part of our state, but we have relatively few collections from that area. And this was why my graduate student, Brianna, was really interested in working on a flora that represented the Chihuahuan Desert. So she's now working in the Broca Mountains. So just showing how, kind of trying to synthesize our collections information can really help to better inform where we can best spend our efforts moving forward. So coming back to the alpine, the alpine is actually down in here. This is showing it a little bit better, kind of at a broader ecosystem scale. So in pink, we have the basin and range, the same comparison, number of collections to area. So we have a lot of collections in the basin and range, but it's also a big area. So that makes sense. And same, so then the Great Plains, right, are almost the worst, right? So a big area, but very few collections represented in our state. Southern Rockies, it's a smaller area. So there is some kind of proportionality here, right? Smaller area, but we still have relatively very few collections. So anyways, this project, we're trying to merge all of our collection information, again, with the spatial, different types of spatial data to try to understand where we've collected and use that information to inform future efforts. I didn't show temporal biases, but we also have some big peaks of collecting efforts in the state of New Mexico. There was a big push in the 1960s. A lot of this had to do with changes in policy and Endangered Species Acts coming out and stuff like that. And then in the early 2000s, there was a lot of collecting from students who are working on floristic projects. So Ron Hartman was a prolific collector in the Rocky Mountains, and he had students in New Mexico doing big inventories. So, big takeaways for looking forward. We need a lot more collections that are recent. Collecting efforts have really dropped off in the last 2 decades. The grasslands and Chihuahuan desert by area, but also the alpine are big, open fields of effort. Tons of collecting has been done around roads. That makes sense, right? But it's one of these things, if we want to have a better, like a more holistic perspective of where plants are, we need to get off the roads a little bit. And then we are hoping to look a little bit more about, different qualities of species, like looking at rare species and maybe other species of interest. Okay, so that's kind of the work that I've been doing, looking at these historical collections. And so very quickly, I wanted to show you what we're doing to add, given now what we know about, where past collection effort have been, how are we trying to improve that? And so I've started this big collaboration with various groups across Western North America. So we're down here in New Mexico, but I have collaborators at Wyoming, up in Idaho, at University of Washington and Western Washington, as well as in British Columbia. And what we're doing is trying to coordinate collection efforts in the alpine. So we're going out to bring back specimens, vouchered specimens from these remote areas, but we're following really very similar collecting strategies so that we can hopefully compare patterns of alpine biodiversity better, okay? And then, of course, the goal in the end is to be able to test some specific questions that we have about what might be shaping these communities. So we're going out to all of these different mountains. So the pink under here is highlighting alpine areas across the Western North America. And these different colored dots are highlighting the summits that we've been to. So there's been a ton of effort up in British Columbia. They have a lot of money from the Canadian government to fly helicopters into these areas and spend a significant amount of time out there. So that's pretty awesome. Maybe someday down here. But we're all going out and collecting vouchered specimens. For the most case, also bringing tissue samples back so we can do some genetic research. We're also collecting geologic samples. As you can imagine, we have lots of ideas about how the substrate is shaping these plant communities. And we're recording a lot of information about the communities themselves. So besides like the typical type of information you record about phenology, we're also trying to understand the abundance of the plants on different summits and where they're found like around the aspect of the summit. So this is kind of a snapshot of what it looks like for us and our teams down in the Southern Rocky Mountains. These are pictures from last summer collecting mostly around Southern Colorado. So, again, these are pretty intensive efforts, backpacking out with presses on your back, spending multiple days out there in the field. And most of this, again, was led by my student, Joseph Kleinkampf, who's over here doing some abundance estimates on top of this summit. But it's a really fun time. We come back and we have all these plants that we get to press, oftentimes in afternoon monsoon storms. So here we're taking shelter at a park up outside of Santa Fe. But yeah, it's been pretty cool experience. So last summer, these are the different peaks that we sampled. Again, mostly around Southern Colorado because there were fires all around New Mexico last summer at the time we were planning to go out. So this year, we're hoping to spend more time in northern Mexico. That we sampled on 8 summits. We brought back 551 voucher collections, representing 182 unique taxa and 34 different plant families, which is like a really incredible diversity, mostly representing our big groups that we saw, the Asteraceae, Poaceae, Brassicaceae, etc. We're also finding new records for the areas. So, in some cases, we, are encountering different species that had never been collected on those mountain summits before. So really just not only adding new information, but in some cases adding new species records, which is really cool. Yeah, so expanding again back across all of the summits, so across the Western North America, mostly Cascades and Rocky Mountains. We sampled 41 summits so far that we have kind of tallied and cleaned our data for. So we're still waiting for some from British Columbia here. This brings our total of alpine taxa in Western North America above 500. And almost 50 different species. Again, a ton of Asteraceae, right? That is a big group in North America. We find that there's only a few species, a few species that are found across all of the summits. For the most part, we find sort of a lot of endemism, or a lot of local endemism, where we see species that are found on only one or a few summits. So that's sort of been a really interesting general biodiversity pattern that we're trying to understand. We don't know why there's such a high turnover across these areas. We're trying to address a lot of different questions that kind of require us to look at species relationships. But one of the advantages of this is that we can actually kind of map out this endemism or kind of biodiversity patterns across space a little bit. So here, these different colors correspond to the different alpine summits. These cooler tones generally are summits that are found in the Southern Rockies. And the warmer tones kind of increasingly go north. So there's a lot of sort of breaks in habitat distributions that we're seeing, which is really interesting. So here's a cool example of Sassafrica bronchialis, where we find it down in the Southern Rocky Mountains, but then not so much going up north and into Idaho, but then almost on every single alpine summit in the Cascades. So just kind of a snapshot of ways that we're looking at all of these data, and we're trying to analyze this further. This kind of shows also broadly speaking, differences in biodiversity patterns across space. So these blue tones here are indicating summits that were sampled that have a higher number of species on them than we would expect, and like species from different families. So really high diversity of distantly related species. Moving down into Idaho and even down in the Southern Rocky Mountains, we see more sort of bursts of families. So we might see like a lot of species representing the Asteraceae, for instance. So kind of a lower overall diversity, even if we have like a lot of species, a high number of species, they're all coming from one or a few families. So this is also a pattern that is popping out that we're interested in exploring more. So getting back to kind of just running through some of these questions that we're interested in, we're trying to understand how the migration of plants over time, like evolutionary time, might be structuring these communities. So we're exploring alternative hypotheses about migration, either from Thuringia, from lowlands, speciation within mountain summits, or residual refugia that are coming from the last glacial ice age. We're comparing these, some species that have sort of a wider distribution but are only found in alpine areas. So they're sort of specialist alpine species like Erigeron humilis with alpine species that are found also at lower elevations, and kind of comparing different aspects of their genetic composition. And finally, we're interested in going back and looking at some of the physical characteristics of these species to try to maybe understand if there are similar mechanisms or modes that these different diverse plants are using to be able to survive, like they're converging on a similar pathway. And also understanding the timing of this evolution of their different traits to see if they evolved before the alpine habitat became an alpine habitat in North America, or if it was something that was kind of forced upon them as maybe they were lifted up as the mountain building happened. Again, trying to understand the big picture, all these bigger picture questions to try to predict how these communities will change over time. We know that in a lot of other places, plant communities are shifting upwards. And we know this again from comparing historical collections information. So this is an example from Mount Chimborazo in Ecuador. In 1802, there was much more alpine habitat, but now lowland species are migrating upwards and outcompeting plants on the tops of the mountains. And this is often called the elevator to extinction, right? We know too, again, looking back at historical records, that snowbanks used to endure throughout the summer on the slopes of New Mexican summits, right? We don't really see that anymore, and there's obviously data to to back this observation up. But this is something that another one of my students is trying to look at. She's comparing herbarium specimens that have been collected over the past 140 years with the current distributions of those species to see if we can detect some of these shifts. So takeaway from but I hope you remember, I guess, from this talk is that, there's a lot of information that we have in these historical collections. We can use them to do a lot of different things to fill in knowledge gaps and inform priorities for future collecting efforts. So these digitization efforts have really revolutionized botany, right? Are really fundamental to understanding biodiversity patterns, and a super powerful tool for addressing the bigger questions we have about ecological and evolutionary processes that are causing all this biodiversity to accumulate, and of course what will happen to it in the future. So with that, I have had, again, a great team. I've already really introduced them, so I don't have to spend much time here on this slide. But my students have benefited from the support of Native Plant Society as well, so I had to mention that. So thanks all for your attention and for coming to my talk today, and I'd be happy to continue this discussion. Yeah.
I have a question about collection methodology over time, and whether the methodology has stayed largely the same, and if it hasn't, how have individual collections sort of indicated what their methodology was. So for example, this is prompted by the slide that you had that showed areas where you seem to have higher concentrations than expected, and in some areas lower concentrations than expected.
Yeah.
And I've been wondering, could that be an artifice of the methodology used?
It's possible, for sure. So we can only map species that are mappable, right? So if there's a collection from 100 years ago that had a very vague indication of where it was collected, oftentimes we have to exclude that because we don't know really where it was. And so that could, remove some of those occurrences and sort of underestimate the species that have been collected there. So that is possible. So it's definitely skewed probably more towards more recent specimens, especially at that scope. You can do a little bit more sort of investigative work to try to figure out where these species were at a smaller scale. So my student Erin is doing that. She's looking at literally every specimen and seeing if she can try to figure out exactly, where it was collected. So yeah, there are definitely issues with that type of thing, especially the spatial stuff. Lots of like other types, like pressing the plants hasn't changed that much, but it's this associated data. So yeah, where it was collected exactly. Elevation is a hard thing. So it's been really hard to record, your altitude until relatively recently too. So yeah.
How about I'm assuming today you collect everything you find.
Yeah.
In the past, was that
Yeah, so that's a really interesting question, because that has there's been trends that have happened through history, right? So kind of at the beginning of westward European expansion, there was a ton of collecting everything because everyone wanted to know what was, what resources there were, in the West. So we do have actually some pretty good, very old collection information in that regard. But then things started shifting towards researching specific plant groups, right? So like a researcher was interested in sitting Asteraceae, for example, and would only go and collect those species. So there is patchiness because of that. So you can usually tell some of that from the collection information itself, right? It will sometimes tell like the scope of the project or something like that. So you can kind of understand that a little bit, but yeah, it's interesting. Yeah.
I have a question. I was very perplexed by the bimodal distribution of openness. And I was wondering if it's, misquoting Jansen, that it's simply because you have the alpine peaks are
harder to
much higher in the tropics.
Yeah.
Is that the explanation?
So that is one of the explanations.
So
yeah, that is one of the hypotheses that we have for that pattern of biodiversity in general. So that pattern,
that's a good title for a paper.
Exactly. Yeah. So it does reflect kind of what has been shown in lots of other groups, like birds, for example, is like one of the classic examples. So that could be it. There's also just differences with the orogeny of so the mountains are totally different histories, and that's something we haven't explored as much to try to see if we can see if there's any kind of effect of that historical process as well. The mountains are tall in the Andes, but they're also really tall here. So there's a lot of different, I think, factors that are going into that. But
the other question is, I've only seen one example, and I'm really curious about alpine networks of pollinators and plants. And the only one I've seen is Pikes Peak.
Yeah.
But are you guys trying to do anything connecting those two? Because there are good collections of these stuff.
For sure.
And maybe I'm calling the legs. I'd be so cool.
I know. That picture the diversity of the Idaho summit that I showed, how it illustrated all of that floristic color and stuff. We've been thinking a lot about that, right? You somehow wouldn't expect there to be so much color in such an extreme environment unless pollinators are really important, right? Otherwise, you would probably see a lot of like wind pollination syndrome plants and not as much showy flowers. And so I have a student that is kind of trying to get at that, an undergrad, who's looking at pollination syndromes of plants to see if he can see if there are differences in sort of potential pollinator communities. And the problem is we have really, really poor collections information on pollinators across this spatial scale. There are, some very well-studied areas like Pikes Peak, the Rocky Mountain Biological Station as well, they've been doing this type of surveys for a really long time. And they've definitely shown interactions, especially over elevation gradients and pollinators and plant communities. But it's a hard thing to get at this scale. So we're kind of trying to address it from looking at pollination syndromes from the plant traits. But
yeah, we'll talk about it later.
Yeah.
I'm very curious about much things.
For sure. Yeah.
So when your diversity indices, did you weight them by area
Of the habitat?
Oh, good question. Yeah, so we haven't across the entire scale yet because we just pulled those data. We're still pulling those data together, but within our different regions, so like within the Rocky Mountains or within the Cascades, we haven't seen much of impact or an effect of either area or maximum elevation with diversity. Yeah, so
little patches are replaced.
yeah, exactly. We think it has a lot to do with the biogeographic history of the species. So where they're migrating from and the species range. And so that's what we're trying to do next, is to try to quantify, how widespread different species are. And if that is just explaining a lot of this overlap. So, yeah.
Are you able to determine or have enough information to answer the question of what global warming is doing to the diversity or health or of alpine species?
That's the goal. So again, we don't have a great baseline understanding of what the communities looked like in the past. We do on a few summits that have been better studied, particularly like in Colorado. There's a few. Pikes Peak is actually one that my students went back to, but in a lot of these places, there's been 0 collections made in the past or a handful that are random. And so it's really challenging to look at from the biodiversity perspective. What we're trying to do is maybe look at a few species that are widespread, that are found across the range, and maybe look at signatures in their DNA that could tell us if the populations have been changing. And so that's one way that we're trying to see if we can understand how more recent climate change might be impacting these communities. So that's one angle that we're taking, but it's hard because we don't have that baseline data. So we're hoping at least now we'll have a snapshot of what it looks like, in the last few years to monitor moving forward as well.
Are you using any drone photography?
Oh, that's a great question. No, a lot of these places you can't fly drones because they're wilderness.
Oh.
And so you can't, that's a big challenge. Yeah. And also with helicopters, it's challenging in North America. You have to hike in. Yeah.
I'm just trying to figure out how elevation and alpine go together. Is alpine in the northern climate in a lower elevation than here in Mexico?
Yes, absolutely. So as you move north from the Rocky Mountains, alpine it's almost like a linear relationship. Once you get to Alaska, you almost step out off of the ocean and there's alpine plants, So yeah, we are at the highest elevation of alpine in North America down here in the Rocky Mountains. So yeah, definitely. But we've looked at maximum elevation and looked to see if that might be driving like the number of species that we're observing in different mountain summits. We don't see that. So we still see a pretty even, we find about between like 50 and 100 species on each summit. Yeah.
So what determines the border between alpine and subalpine?
Yeah, it's dictated by the tree line, right? Of course. And so physiological tolerance of these trees, but we still have a tree line that's very high down here in the Southern Rockies, and in Colorado is still very extreme climate. And so, that's a question.
Competing the alpine.
Not just the tree. I mean, it's hard to say which is coming first too. And there has been some work on that. There's a lot with soil microbes as well. And I haven't even touched the below ground stuff. But a lot of people have looked at that and are trying to understand, for instance, if like the soils are changing first and then the plant communities migrate following. So it's like, which is happening first is another question. Yeah, that's definitely more on like the like ecological, like on the ground type of stuff, but yeah, experimental.
A great talk.
Thank you. Thank you all.
One question from
Oh, yeah. before the
Wendy and Don Graves asked, are you noticing any possible extinctions as you compare old collections with newer collections?
Oh, yeah, that's also, it's a local extinction within a mountain summit. That's again, hard to determine. There are some cases that we think it might have happened. But that type of thing would be something that in order to say with confidence, we probably would want to resample after a couple of years, because sometimes you can have just years where things don't show up. Not so much in the alpine because so many things are perennial. So it's not as big of a problem, but there's definitely cases in other ecosystems where that's happened. Also cases where, plants have gone extinct completely, like the species is extinct, but we have herbarium records representing those species. So it's just kind of crazy. Yeah.
And one more question. You had professors who were very instrumental in your career path. Can you speak to the value of science education in encouraging youth to be excited in our native ecosystem?
How long do you have? Yes, of course I can speak to that. I mean, it's essential. I think it's getting better. I think there's, a lot of, social media effort and stuff like that. And I think iNaturalist has done wonders, really, like bio blitzes and these types of things. I think COVID, one of, I think the positive things that came out of that was people started looking in their backyard at things and trying to, I have a lot of students who were like, I was trapped inside and staring at my plant all day and now I want to learn what it is. And so that, like students like that. So yeah, educate is super important. It's a hard thing, but it's just a very challenging, field, but it's definitely important. So yeah.
Thank you very much.
Thank you all. Can I just stop sharing or leave it?
We can stop sharing and stop recording.
And I hope you all
Bye, everyone at
join
home.
us for some
So I need to talk with you briefly,
Sure.
because I care deeply about